Hello everyone, welcome to today's presentation. I'm excited to introduce our topic, heredity and genetics. In this session, we'll explore how traits are passed down from one generation to the next and the fascinating science behind it all. We'll cover key concepts, examples, and the importance of genetics in our lives. Let's get started. Let's now look at slide 2, which covers some fundamental concepts in genetics. First, we start with heredity, which is the passing of traits from parents to offspring. This leads us to the accumulation of variations during reproduction, explaining how genetic diversity is maintained. Moving on, one of Mendel's key contributions was his work on inheritance, where he studied how traits are inherited through specific rules. These rules include the law of segregation, which states that alleles for a gene separate during gamete formation, and the law of independent assortment, which explains how genes for different traits are inherited independently of each other. Next, we have genes and alleles. Genes are units of heredity, and alleles are different versions of those genes. Mendel's laws are built on understanding how alleles behave during inheritance. For example, the law of dominance explains how certain alleles mask others in heterozygous conditions. When we talk about genotype and phenotype, we're referring to the genetic makeup and the observable traits, respectively. In the context of inheritance, knowing whether an individual is homozygous or heterozygous helps us understand their genetic composition. And when dealing with the inheritance of a single gene, these principles become even clearer. All of these concepts tie together to give us a comprehensive picture of how traits are passed down and how genetic variation arises in humans. Today, we're going to talk about a fundamental concept in biology, heredity. Heredity is the process by which characters or traits are transferred from parents to their offspring. Essentially, it's how certain features, like eye color or height, are passed down through generations. When we say characters or traits, we're referring to the observable features or characteristics that make each organism unique. This transfer of traits is what connects parents and their children at a biological level. Understanding heredity helps us see how traits are inherited and why organisms are similar to their parents. It's a key piece of the puzzle in understanding how life is passed from one generation to the next, and it ties into the larger theme of how genetic information shapes living beings. Now, let's talk about variations. These are the differences you see between individuals of the same species. Essentially, variations refer to the unique traits or characteristics that set one individual apart from another. They can be things like eye color, height, or even certain behaviors. Recognizing these differences is important because it helps us understand how species adapt and survive in different environments. Throughout this presentation, we'll see how such variations contribute to the diversity of life and play a key role in evolution. So, keep in mind that variations are the natural differences among individuals that make each one special. Some variations that occur during reproduction can actually be quite helpful for an organism. They allow it to adapt to changes in its environment, which is essential for survival. But on the other hand, not all variations are beneficial. Some can be neutral or even harmful, and if these harmful variations accumulate, they might lead to the organism's extinction. When organisms reproduce, whether sexually or asexually, minor differences or variations often appear in their offspring. These variations mainly happen because DNA copying isn't always perfect. In asexual reproduction, since the offspring are essentially clones of the parent, the variations tend to be fewer. But in sexual reproduction, because of the combination of genetic material from two parents, there's a higher chance of variation. This accumulation of variations during reproduction is a key driver of evolution. It's what provides the raw material for natural selection to act upon, helping species adapt over time. So, understanding how these variations build up and their effects is crucial for grasping how life on Earth changes and evolves. So, Gregor Johann Mendel, a pioneering scientist, carried out experiments with garden pea plants that really laid the groundwork for our understanding of inheritance. Through these experiments, he was able to identify the fundamental rules that govern how traits are passed from one generation to the next. Now, one of the key ideas here is that the inheritance of characters, that is, traits, happens because both the father and the mother contribute equally to the genetic makeup of their offspring. This means that each parent provides a set of genetic material, and together, they influence the traits of the child. For each trait, there are actually two factors involved, one inherited from the father and one from the mother. These factors are what we now call genes, and they determine the specific characteristics that appear. These characters are transferred through genes, which are present in DNA molecules. And these DNA molecules are packed into chromosomes, which are located in the nucleus of the cell. This is the cellular basis of inheritance that underpins everything Mendel discovered. So, understanding these principles helps us see how traits are inherited and how genetic information is passed down in a predictable way, which is fundamental to the overall theme of genetics and heredity. Let's take a closer look at this slide, which introduces some key genetic concepts. Here, we see the idea of traits, which are specific characteristics like plant height or flower color. Each trait has different versions known as alleles. For example, in this diagram, T represents a dominant allele, and T would be the recessive allele. When an organism has one dominant allele, like T, that trait shows up. But if it has two recessive alleles, like T and T, then the recessive trait is expressed. This slide also highlights that some traits are dominant, meaning they mask the effect of recessive traits when present. Recessive traits only appear when an organism inherits two copies of the recessive allele. Understanding this distinction is fundamental to Mendel's work on inheritance, which we'll explore further. These concepts form the foundation for how traits are passed from one generation to the next, helping us understand the inheritance patterns we observe in many organisms. Now let's look at how crossing plants with contrasting characteristics works. When we cross two plants that differ in a specific trait, all the first generation, or F1, plants end up showing just one of the parental traits. This suggests that the trait from one parent is dominant over the other. This process highlights the inheritance of a single gene, which plays a key role in how traits are passed down. The fact that all F1 plants express the same trait points to the idea of dominant and recessive alleles, where the dominant allele masks the effect of the recessive one in heterozygous conditions. Understanding this helps us grasp the basics of inheritance and why certain traits appear in some generations and not others. It's a fundamental step in unraveling how genes control the traits we see in plants and in all living organisms. Now let's talk about self-pollination, which is the second step in Mendel's experiment. After crossing the plants, the F1 plants are allowed to self-pollinate. This means they pollinate themselves, which helps us see how traits are passed down within the same genetic line. What's really interesting here is that the traits we see, specifically the dominant and recessive traits, appear in a specific pattern. In this case, they show up in a ratio of 3:1. So 3 parts show the dominant trait, and 1 part shows the recessive trait. This ratio is a key clue to understanding how traits are inherited and how dominant traits tend to overshadow recessive ones in the next generation. It's such a fundamental concept that helps us understand the basics of genetics and inheritance patterns. Let's take a look at the F2 generation and the F1 generation. When we talk about the F1 generation, we're referring to the first set of offspring resulting from the original parental cross. These are often uniform because of the dominant traits expressed. Moving on to the F2 generation, which is produced by self-pollinating or crossing the F1 individuals, we start to see more variation and segregation of traits. This pattern helps us understand how traits are inherited and how genetic variation is maintained. Finally, in our conclusions, we tie everything together. We've seen how the F1 generation demonstrates the dominance of certain alleles, while the F2 generation reveals the underlying segregation patterns. These insights are crucial because they form the basis of Mendelian genetics and help us predict inheritance outcomes. Overall, understanding these generations allows us to better grasp how traits are passed down, which is central to our broader discussion on genetics and heredity. So, Mendel proposed that there is something being passed down in a stable way from parents to their offspring. This something remains unchanged as it moves through generations. He believed that these passing units are contained within gametes, which are the sperm and egg cells. Mendel called these units factors, but today we know them as genes. This idea was really groundbreaking because it laid the foundation for understanding inheritance. His conclusions showed us that traits are inherited in a predictable way, which is essential for understanding genetics as a whole. This concept connects directly to our overall theme of how traits are passed and why we see the diversity we do in living organisms. Let's talk about alleles. Alleles are essentially different versions of the same gene. They are the variations that can exist at a specific spot on a chromosome. Think of them as different options or flavors for a particular trait. For example, the gene for eye color might have alleles for blue, brown, or green eyes. These variations are crucial because they contribute to the genetic diversity we see in populations. When we look at an individual, they inherit one allele from each parent for every gene, which together determine their traits. Understanding alleles helps us grasp how traits are inherited and how genetic variation drives evolution. Overall, alleles are fundamental to the study of genetics because they explain the differences we observe in living organisms. Now, let's talk about different forms of the same gene, which are called alleles. Alleles are essentially variations of a gene that can exist in a population. Genes themselves are the basic units of inheritance. They carry the information that determines specific traits in an organism. When we look at genes that code for opposing characteristics, like tall versus short plants or blue versus brown eyes, they are known as alleles. Understanding alleles helps us see how genetic diversity contributes to the variety of traits we observe in living organisms. This concept ties into the larger theme of inheritance and how traits are passed down through generations. Now let's talk about homozygous and heterozygous conditions. When we say an organism is homozygous, it means that both alleles of a gene are similar. For example, if both alleles are dominant, like TT, or both are recessive, like TT, the organism is homozygous. On the other hand, if the two alleles are different, such as TT, then the organism is heterozygous. Heterozygous conditions are interesting because they often determine how traits are expressed, especially when it comes to dominant and recessive traits. For instance, in our example, TT is a heterozygous condition, which results in the tall character because the dominant allele T is present. Understanding these conditions helps us grasp how traits are inherited and expressed in offspring. The key takeaway is that homozygous involves two similar alleles, and heterozygous involves two different alleles. This distinction is fundamental when studying genetic inheritance and predicting traits in future generations. Let's look at some examples. TT, TT, TT. These represent different genetic combinations or genotypes. The genetic makeup of a trait, which is the specific set of genes an organism inherits, is called the genotype. For instance, tall or dwarf plants are examples of traits that are determined by their genotype. Now, moving on to the observable aspects of these traits, which are what we can actually see or measure. These are called phenotypes. Think of phenotypes as the descriptive features or characteristics, like height, flower color, or leaf shape. They are the outward expression of the genotype. Understanding the difference between phenotype and genotype is fundamental because it helps us connect the genetic basis of traits to what we observe in real life. This distinction is crucial in genetics, plant breeding, and many biological studies. Now, let's look at the ratio observed in this F2 generation, which was 9:3:3:1. This ratio is very significant because it reflects the underlying principles of inheritance. When these plants were self-pollinated, out of every 16 plants, 9 showed round yellow seeds, which we denote as RRY. 3 had round green seeds, RY, 3 had wrinkled yellow seeds, RRY, and just 1 had wrinkled green seeds, RY. This pattern isn't random. It's a clear indication of how two traits, seed shape and color, segregate independently. Mendel, focusing on these two characteristics, selected pea plants with specific seed traits, one with round yellow seeds, which is RRYY, and another with wrinkled green seeds, RY. He then cross-pollinated these plants, and interestingly, all the F1 plants showed round yellow seeds, which are RR Yai. This is a key point. The F1 generation was uniform, showing the dominant traits. Now, this setup is what we call a dihybrid cross. It's a crossing involving two traits at the same time. This experiment allowed Mendel to observe how these traits are inherited together and how they segregate during gamete formation. Understanding this ratio and the inheritance pattern helps us grasp the fundamental principles of genetics, how traits are passed from one generation to the next, and how dominant and recessive traits behave in a predictable manner. It's a cornerstone concept that unlocks the mysteries of heredity. Let's take a closer look at Mendel's laws. First, we have the law of dominance, which explains how in a pair of alleles one can mask the presence of the other. This is why, for example, dominant traits appear more frequently in offspring. Next are the first two laws derived from the monohybrid cross. The law of segregation states that during the formation of gametes, the two alleles for a trait separate, so each gamete carries only one allele. The law of independent assortment, on the other hand, tells us that genes for different traits are inherited independently of each other, which is evident in dihybrid crosses. These two laws highlight the fundamental principles of how traits are inherited and help us understand the patterns we see in genetics. Overall, these laws form the foundation for understanding inheritance and genetic variation. So on this slide, we're talking about the law of dominance. Essentially, it states that the dominant character is expressed in the F1 generation. That means, when we cross two different traits, the dominant one will show up in the first generation. Now, in the F2 generation, after self-crossing those F1 plants or individuals, we see both the dominant and recessive characters expressed. And interestingly, this follows a 3:1 ratio. So, for every 3 individuals showing the dominant trait, there's one showing the recessive trait. This pattern really highlights how dominant and recessive genes behave when passed down through generations. It's a fundamental concept that helps explain why traits appear in predictable ratios, which ties back to the overall theme of inheritance and how genes are transmitted from one generation to the next. Now let's discuss the law of segregation, which is a fundamental principle in genetics. Essentially, it states that the two alleles of a trait, remember, these are the different versions of a gene, segregate from each other during the formation of gametes. What this means is that each gamete, like a sperm or egg, receives only one of the two possible alleles. So, if an individual has two different alleles for a trait, only one will end up in each gamete. This segregation ensures that when gametes fuse during fertilization, the offspring have a predictable combination of alleles. It's a key concept because it explains how genetic variation is maintained and passed down through generations. This segregation process is a cornerstone of Mendelian inheritance, helping us understand how traits are inherited in predictable patterns. So this slide introduces the law of independent assortment, which is a fundamental principle in genetics. Essentially, it states that the alleles of one trait, meaning the different versions of a gene, segregate into the gametes independently of the alleles of other traits. In simpler terms, how one trait sort of splits and gets passed on doesn't influence how another trait does. This is a key concept because it helps explain why we see such a wide variety of genetic combinations in offspring. It emphasizes that each trait is inherited independently, contributing to genetic diversity. Connecting this to the overall theme, understanding this law is crucial because it underpins how traits are inherited and how variation arises in populations. It also sets the stage for predicting genetic outcomes in more complex crosses. So, in essence, the law of independent assortment helps us understand the randomness and independence in the inheritance of different traits. When a sperm with an X chromosome fuses with an egg that also has an X chromosome, the result will be a girl. Conversely, if a sperm carrying a Y chromosome fuses with an X chromosome egg, the resulting baby will be a boy. It's important to remember that both sperms and eggs carry just one set of sex chromosomes, and all eggs always carry an X chromosome. The key difference is in the sperm. Some carry X, others carry Y. Humans have a total of 23 pairs of chromosomes inside each cell's nucleus, and among these pairs are the sex chromosomes. X and Y females have 2 X chromosomes, making their pattern XX, while males have 1 X and 1 Y, making their pattern XY. This process is what determines the sex of the baby, and it highlights how the combination of chromosomes from each parent influences whether the child will be a boy or a girl. It's a simple yet fascinating mechanism that connects to the broader theme of genetics and inheritance.